Thank you. Good afternoon. So I'm going to talk a little bit about gene therapy for GAMP deficiency. And I think we all know this already. Again, GAMP deficiency, guanidinoacetic acid is elevated, creatine is depressed. And Olivier already discussed adeno-associated viral vectors, which we've employed in our gene therapy approach. In this approach, essentially all the genes from the virus are removed and replaced with what we call a transgene cassette that has regulatory components that then cause expression of the GAMP gene by this virus. So I'm just going to jump right into the data here. So we made a virus that we could administer intravenously, whether it be into a human or in this case into a mouse model, into a vein in the arm. It doesn't have to be injected into the brain, into the fluid around the brain. It can be injected straight into a vein in the arm, into the leg, in another location. And in this viral vector, we used a liver-specific promoter. So this gene really only expresses in the liver by this virus. We used a special type of viral coating called RH10, and we had some other regulatory elements that increase expression. So here's some sections of liver. On your left side is actually without the GMET staining, but the RNA, the genetic component that leads to the protein. In pink, you can see this here. RNA is unstable, it is eventually lost. But over here, you see the brown color is the staining of the liver cells. And what you can see here is probably roughly about 90% of the cells are now expressing the protein from a single injection of this viral vector when this mouse was euthanized 1 month after the injection. So with this dose of virus, 1 × 10^14 genome copies per kilogram, we really transduce or have expression within a very large number of liver cells. Now, what does this result in? So I have the data presented a certain way here so you can see changes with dose administration. A key component to viral-based gene therapy for humans will be the dose that you will eventually receive or is administered to the patient. A lower dose is safer than a higher dose. So on that side of the screen, on the upper graph, is creatine. This is measured in the blood. On the lower right side here is guanidinoacetic acid. We start with a lower dose and move across. So each mouse is serving as its own control. So the mouse gets bled, it's then given the virus, and then we check in the blood a month later what the level of creatine is and what the level of guanidinoacetic acid is. So in red is before, and in blue is after. So when we give the lowest dose, 5 × 10^12, so that's 5 with 12 zeros afterwards per kilogram. Thank you very much for this. What you see is before we have very little creatine, but then afterwards the creatine increases. As we increase the dose, 1 × 10^13, the dose and the level in the plasma of creatine goes up. When we increase times 5 × 10^13, kind of a more desirable dose, we get this type of level, near normal level of creatine in the blood. And then at the highest dose, 1 × 10^14, we reach what is essentially normal, a normal level, 30 days after the injection. With regards to guanidinoacetic acid, we see an opposite effect. Of course, the desire is to have the GAA go down. So before, we see in red here what it is in the blood. And then we give the virus, 30 days later, we measure, it declines. We try in a different group of mice, we increase the dose, and we see it decline. We increase the dose again to 5 × 10^13, we see it decline. And then when we eventually give the highest dose, we see that guanidinoacetic acid is completely resolved in the plasma. So we see this dose effect. And in part, we do this to assess what might be the dose we need to give in people eventually, if this were to go to clinical trial. So we see that with this viral vector we express in the liver, in the plasma, that is in the blood, we can have creatine levels be normal, we can have guanidinoacetic acid levels be resolved, that is, be gone completely. Now, we these mice were developed by Dirk Isbrand when I think he was at Hamburg, who's now at the University of Cologne. And like the SLC mice, they're a little bit smaller. They're not as small as the SLC mice, but this is a knockout mouse here. That is a mouse that has GAMP deficiency. You see in the center a wild-type mouse, a littermate, normal-sized mouse. And then the treated mouse, a year after the administration of the viral vector, the size is almost equivalent. It's a little bit smaller, but it's almost equivalent. But the quantitative data is above. So we'll start with the females. So in red, red here is the knockout mice untreated for all the data that I'm going to show to you. Over that year, the mice were injected when they're 2 months old. The mice really gain very little weight. They're flat. But the mice that are normal, the wild types, are in black, and we see the weight increase over that year. And we see in the females that in blue, the treated mice that have GAMP deficiency but treated with this viral vector have a near equivalent weight gain as the wild type mice. In the males, we have a slightly different picture. That is, the knockout mice, the GAMPed untreated mice in red, don't really gain much weight over the year, but as the wild types do in black. And then the treated mice, the GAMPed knockouts treated with the viral vector, gain some weight, but don't reach equivalency. But they gain a substantial amount of weight over that period of time. Now, I showed you earlier just a dose response. We gave different doses. We saw the response, that is, the creatine went up in the plasma, the GAA went down. But what happens over time? What happens over that year? So let's start with the upper graphs, and we look at the females. So in the top two graphs here is guanidinoacetic acid. So in red, if we start with the females, untreated mice, GAMPed untreated mice, there's some variability when you do the testing. The mice have some different responses, but these untreated mice, you see that the guanidinoacetic acid is high, it's elevated. But we see in our wild-type mice in black, it's very low, guanidinoacetic acid the whole time. Once we give the injection of the viral vector in blue, the first time we measure 1 month later, the guanidinoacetic acid is completely normal and stays that way for an entire year. We see a very similar picture in the males, really no difference. If we look at the creatine levels over here, starting with the females, the creatine levels in the untreated GAMP deficient mice are very low in the plasma, that is in the blood, for that entire year. In the wild-type mice in black, we see the level here. In our treated mice, we see at the first time point that we measure the creatine level in the blood, in the plasma, is completely normalized with this dose, 1 × 10⁻⁴ genome copies per kilogram. We see a near identical picture in the male. So what I've shown you to this point is that over a year, and typical mice in the lab live about 2 years. So this is about half their lifespan. That's where we stopped the study. We did not continue beyond that point. But we see that the level in the blood is normal. Now, we also measured this in the urine. So here the males and females are together. They're not separated. But if we look at the guanidinoacetic acid, it spills into the urine. It goes out through the tubules. It ends up in the urine. So we see that in untreated GAMP-deficient mice, the guanidinoacetic acid is elevated. And similar to what we saw in the blood, the wild-type mice, GAA is almost non-existent in the urine. And then for the treated mice, the mice were injected at time point 0, and we didn't check the urine as frequent. We checked it at 3 months, and we see it's normal in the males and females. And same with the creatine, the same type of pattern. In the knockout mice, there's no creatine in the urine. In our wild-type mice in black, we see there's creatine in the urine, and then with the treated mice, it's similar to the wild-type mice. So the therapy to this point is looking in the plasma is very effective. Looking at the transduction, or how much virus gets into the liver, is very effective. Now, at a year, we euthanized those mice, and we took out different organs, different tissues, and we analyzed them for the creatine levels and the guanidinoacetic acid. It's great to correct it in the plasma, but we also know that guanidinoacetic acid can be produced in some tissues, so it's important to understand what's going on in the tissues or the organs themselves. So first, if we look down here, these are liver enzymes. So this is first just to examine whether there's toxicity from giving this dose of virus to the mice. These are tests that maybe your children have had, maybe you have had, and certainly patients in clinical trials with AAV have, because most of the virus, even in, if you give a virus for SLC6A8 in the bloodstream, most of it's going to go to the liver. It just ends up going there for different reasons, part of it being filtration, part of it for blood flow patterns. But if we look at the AST, a liver enzyme, and the ALT, and we compare the wild type in black with the treated mice, there's no difference in those liver enzymes. It would be concerning if they were elevated. In the GAMP deficient mice, the enzymes are slightly elevated, but I would not call these numbers, 30 to 80, being elevated transaminases. They're essentially normal. They're essentially normal in the knockout mice untreated, but they're most importantly normal in the mice that are treated. If we then look at the individual organs, if we look at the liver, particularly the guanidinoacetic acid, we see here in the GAMP deficient mice in red, the guanidinoacetic acid is high, but it's normalized in our treated mice. And we see this throughout. We see this in the skeletal muscle, we see this in the heart. But the two places we don't see it completely is if we look in the kidney, we see that the guanidinoacetic acid level remains a little bit elevated, and we see it remains in the brain at about 40 to 50%. So there is some persistence of guanidinoacetic acid in the brain. And I believe that likely the reason for this is that, as I mentioned at the beginning, this viral vector primarily only expressed in the liver by the way it was designed. But even in that case, we did some additional studies to understand brain function in this mouse model. And when this mouse was originally described, the ISBAN group at Cologne now didn't really find any behavioral abnormalities in this mouse model. If there was any, they were very subtle. In our group at UCLA in Los Angeles, we did some additional studies and were able to find some subtle abnormalities, which I'm going to show you in just a moment also. But here's some PET CT imaging, which has been talked about a little bit as potential biomarker compared with, I think, MRI was that discussion. But what this looks at is glucose metabolism. So if you can imagine that if you don't have creatine or very little creatine in a tissue, you rely on the other source of energy, that being glucose. And what we see in wild-type mice here, this is the normal pattern. So more activity is going to be it's a rainbow, is in red, and lesser activity is in blue. So if we look at the wild-type mouse, the one that I have the laser pointer on, we see much more green, yellow, some red. If we look at the GAMP deficient mouse in the middle here, we see in the brain, which is in that circle, dotted circle, there's much less activity of glucose in the brain. It's blue. But with the gene therapy, again, from the liver, in the liver, we see really a recovery of this activity in the brain of glucose metabolism. This is represented over here graphically. When we studied the mice similarly and just gave them a regular chow, regular food here, creatine supplement and regular chow, and gene therapy was better than regular chow, and certainly better than regular chow with creatine supplementation, was better at improving brain metabolism. So the gene therapy was most effective over creatine supplementation. Now I mentioned to you that in the studies that were done in development of this mouse, there really were only subtle, if any, brain abnormalities. But we, in studying them, came up with, found there was another abnormality in something called the Barnes maze. And what the Barnes maze is this big circle, a disc, and there's these holes in it around that disc. That is represented by the circles that you might see in this disc. This is a diagram of the disc, but each one of these circles here is a hole. And one of those holes on the bottom, each one's just a hole, but on one of them there's a cup. And so the mice don't like to be exposed. So they'll wander and try to find that one that's a cup so they can go into it to hide. And mice can learn just like people. They can learn different ways. They can randomly go from hole to hole to hole, just searching around in a random pattern to find that spot. They can serially walk from hole to hole to hole till they find it. Or when they learn, they know where it is because the disc is in the same place in the room. You don't move it around. They know I start here and it's here, so I go there every time. So what we can see is a pattern develop for these mice. And the lettering's a little bit small, okay? So the wild type on each one is represented on the left. The untreated GAMP deficient mice are in the middle, and the treated mice are on this side, the right side, okay? So wild type, knockout, treated. And what you see over time is the GAMP deficient mice that are untreated don't learn too well. They learn, but they seem to forget. If we look here, this is 1 week after they've been taught. The wild type mice, again, the column on this side, show primarily in blue the direct spatial. They know where it is, they go there every time. And the orange, the serial, they hunt. They don't have this random dissociated pattern. The GAMP deficient mice in the center have the serial, they're searching each time, and have just the remainder part is random. But the treated mice on the side closest to me, where the arrow is they're very similar to the wild type mice. Most of their activity is serial, and some of it is direct. They've lost this random pattern. So they've learned to find the spot to hide over time. Now, one of the other discoveries that we found with these mice is we suspected that they might be ataxic, that is, had some abnormality in walking. But we couldn't really demonstrate that by examining the mice in any studies that we did. But we did find that they had grip strength abnormalities. So the mice are put down over a rod, and they will normally grab onto it, and then you'll pull them off. And this is done in a blinded way by people that are not working with me, so they don't know which group the mice are in. So they can be they're objective, they're blinded, they don't know what group they're in. So when we do this with the forepaws, the front paws, we see in the GAMP deficient mice, their force that it takes to pull them off is less than it is in the wild type mice, but with the gene therapy, it's recovered. It's very similar to the wild type mice, and similar with their hind legs. So we found that we have really an effective therapy that seems to work if we express in the liver, but we do leave a little bit of guanidinoacetic acid in the brain. So now we've embarked on trying to improve our approach. And we've developed 4 different viruses, all again based in that same category, the AAV that Olivier spoke about a few moments ago. But we've modified them in some way. So this is our first vector that expresses just in the liver, and we had this coating called RH10 on the outside. We've modified it to one that works even better in the liver called serotype 8. And we made a completely new virus here that expresses a different type of promoter, a different type of regulatory element that expresses in all these different tissues. And then in the last one here, we've had this a similar promoter, but we've changed the GAMP gene. We've tried to improve its expression by making some changes within it itself. And we've started to study these again as our second generation approach. And we're looking at each one to see how much they might change in the plasma, the creatine levels on the top here. So again, there can be some batch effect on when the assays are run, but we see the creatine level in the plasma in the untreated mice is very low. But in this, our old vector, we see it increases. And with all these other vectors, it increases, except for this last one where we tried to improve the gene itself. Didn't seem to really improve it. But we see that the different vectors work similarly to the first vector that I described. And similarly with the GAA levels, we can continue to cause them to be completely, essentially eliminated in the blood with these new vectors, except for, again, the one that we tried to make an improved gene itself. And we looked in the brain to see now if we could get rid of that guanidinoacetic acid that I told you was about 40% was still there. And in fact, we can with these new vectors. If we look in the center graph here, particularly at this one called AAV9, CAG, the amount of GAA is greatly reduced compared to the animals that are not treated and over our other vectors that we had developed here. In addition, there are thought to be other compounds, at least in mice, in addition to GAA, that are related to it that exist in the brain that may be causing injury, one called GBA, that is also resolved with our new therapy, our new viral vector. The last little area I just want to touch upon is, I mentioned that we suspected that the mice have some ataxia, some different difficulty with motion or movement that we can't pick up on the most sophisticated activity assay of the mice. But again, we suspected that there was something going on because of their grip strength alteration. So sometimes you can take a mouse and you can euthanize it very quickly and take out its brain, and you can put electrodes into certain cells in the brain. Well, there's one method where you can study this cerebellum, the movement area of the brain, which is very metabolically active. The neurons continuously fire at different rates depending on activity and other factors. And we've been looking at this called the Purkinje cell firing in the brains of these mice. And this we thought might help us understand if they actually are ataxic. And normally, if you put a mice on a device called a rotorod, which is basically a rod that's spinning, slowly spinning, a rod like a dowel, it's just rotating, but you have these planes between them, so you can put several mice on there at the same time, and they don't interact with each other, and you can measure when they fall off. And if you have ataxia, difficulty with motion, they would fall off, like on this other side here, some deficit. And we haven't been able to detect that, but we thought, we haven't found that in these mice, but we thought there might be an abnormality still in the cerebellum. And in fact, there is, and we're going to be doing this in these studies on these mice that I've just described with our next generation vectors, where you can take the mice, they're euthanized very quickly, the brain's taken out, and you can put an electrode in a certain location in the cerebellum, and you can measure the spontaneous Purkinje neuron firing. And if we look at the firing frequency, again, these cells just fire all the time. If we look here at the wild-type mice, they have a frequency, this is their pattern. When you measure them multiple times, you can measure the amount frequency of different neurons over time, that we see that the wild-type average rate is about 54, but in the GAMP-deficient mice, it's about 30. So it's going to be an objective measure that we can use to further understand what's going on in the brain, but also to try to see if the gene therapy, another objective measure, has an improvement in presumably this ataxia that also exists that we can only really detect on an electrophysiologic level in the brain. With that said, these are my conclusions for today, that GAMPed mice treated with a liver-specific promoter AAV vector thrive. They're healthy, they're long-lived. There's sustained expression in the liver to at least 1 year of life. There's restoration of plasma creatine levels and resolution of the hyperguanidinoacetic acidemia that occurs. The urine creatine and GAA are normalized. Brain creatine levels are restored. However, there is some brain GAA that remains. However, it's markedly reduced. Looking at FDG PET CT scanning, glucose consumption in the brain is normalized. Learning and motor abnormalities in the GAMP-deficient mice are resolved with this gene therapy. We've developed these new vectors, particularly one based on AAV9 serotype that results in a further brain reduction of GAA and a complete resolution of this compound called GBA. And now we're going to explore this cerebellar Purkinje cell firing as an objective measure further to examine brain function with these new vectors. I'd like to thank my group. I'd also like to particularly thank Andreas at University of Toronto at Sick Kids, and his associate Ilona Takikova, who's done a lot of work with us in these studies. Thank you very much.